handed me the business card of one of the editors at University of Hawaii Press, and said she wasn't able to make it tonight, but wanted to let you know that the press was interested in publishing a book on our work if we were interested. And so it was a very memorable talk for me. I contacted them afterwards, and the book came out at the end of 2016. I have a copy up here if you're interested, and also some brochures from the press on how to order it, and they're giving a discount for ordering it if you want to do it through them. And also, if any of you guys are publishers, that I would like to write, and I'm looking for a publisher. Now, before I get into the talk, I want to mention that the work I'm going to talk about tonight is really a team effort. There's a lot of folks who play extremely important roles in the field, helping collect the information we're collecting, tagging animals, getting photographs, collecting biopsy samples. There's a lot of folks that play an important role in the office, going through thousands and thousands and thousands of photographs. We just finished a 12-day field project in Hawaii, which took 38,000 photographs. It takes a lot of time and effort to go through those. We have a number of folks who play a big role in terms of analyzing data sets, tag data sets, photo ID data sets, and various collaborators who are working with the genetic samples that we're collecting. So it really is a huge group of people that I'm going to summarize a lot of those results tonight. We also have a lot of citizen science contributions, including some folks who are here in this room, folks who are working on the water or playing on the water who take photos and contribute them to our photo ID catalogs, and that plays a big role, and I'm going to talk about some of those results. And this is a long-term study. We've had funding from a lot of different sources over the years. NOAA Fisheries funds a lot of our work, in part because it has a strong conservation and management implications, so it's something they've been interested in. Some of the work we've been doing has been looking at what species are found on the Navy range off of Hawaii, and how different species respond to Navy sonar. So we've gotten a lot of funding from the U.S. Navy, and then groups like Dolphin and various nonprofit foundations have also contributed over the years. This is sort of my advertisement for the citizen science part of things. I can't overemphasize how important photos are from all of the folks who are on the water in Hawaii. We come out to Hawaii for field projects 2 or 3 or 4 times a year for a few weeks at a time. But everyone who's on the water year-round really plays an important role. And you know, whether it's fishermen, and we have a grant right now to buy cameras for fishermen, or tour operators, or photographers, or just weekend boaters. If anyone takes a camera out with them, there's really a lot of opportunities to contribute. Now, in terms of putting Hawaii and our work into context, the Hawaiian archipelago is the most isolated archipelago in the world. This blue line around the archipelago shows the 200 nautical mile U.S. exclusive economic zone. So everything inside that line is U.S. waters, and everything outside is international waters. And this isolation is important when you think about the different species of dolphins and whales that are found here, and whether or not they came from, continental populations or originated from open ocean populations. Now, if you look at it from an oceanographic perspective, this is an ocean color map which reflects productivity, chlorophyll A. Chlorophyll A is produced by phytoplankton, and phytoplankton are fed on by zooplankton, and zooplankton have their predators, and their predators have predators, and up the food web. The bright red areas are the most productive areas up in the high latitude areas and along the continental slopes. And if you look at where Hawaii is in this middle of a very blue area, which means very low productivity. The reason why the water is so clear out here is because of this low productivity. There's not a lot of phytoplankton, there's not a lot of zooplankton. Now, if you zoom into the archipelago, up north there's this very productive area, and if you look into the northwestern Hawaiian Islands, there's a real high productivity signal around some of the atolls and islands up there. Around the main Hawaiian Islands, there's not much going on. And but if you squint, you can see that there is a little bit of an increase in productivity around the main Hawaiian Islands, and it's surrounded by this biological desert. So what that increase in productivity does is it basically creates a small oasis in the middle of this oceanic desert. And that oasis is really important in terms of what species of whales and dolphins are found here and why they're found here and why they persist here. Now the oasis is created by a number of different factors. And if you live on the windward side of the island, you know there's a lot of rain. That rain washes a lot of nutrients into the water, and the nutrients are used by phytoplankton for growing. The winds come from the east primarily and go to the west, and those winds drive ocean currents, they drive eddies, and they sweep all the nutrients that come in from rain runoff into the western sides of the islands, the leeward sides of the islands. The currents, deep water currents basically hits the islands from the east, rise up along the island slopes and bring colder, nutrient-rich water up. These winds drive a lot of eddies that spin primarily off the west side of the islands. Sometimes they're upwelling eddies, like that one that you can see, the counterclockwise eddy off the north end of the Big Island. Sometimes they're downwelling eddies, like the big clockwise one off the south end of the Big Island. Those eddies provide concentrations of phytoplankton and zooplankton, and they basically provide these little microhabitats that are important for a lot of different species of whales and dolphins. Now, I first started working in Hawaii 22 years ago. When I came, I originally moved to Maui. When I moved there, all the researchers, with one exception in Hawaii, that were studying whales were studying humpback whales. And all the researchers who were studying dolphins were studying spinner dolphins. So I didn't really want to overlap with them or compete with them. So instead of studying either humpback whales or spinner dolphins, I focused on some of the lesser known species. Up on the upper left, that included bottlenose dolphins. Upper right, false killer whales. Lower right, pilot whales. And lower left, pantropical spotted dolphin. And these are all species that you can either see between Maui and Lanai in the case of the upper ones and the spotted dolphin, or pilot whales once you got into the deeper waters west of Lanai. So started up photo identification catalogs of those species and working with them right from the beginning. Now, most of my work has been on relatively small boats. This is sort of the typical size of boat that we're using in our work right now. It's a 24-foot rigid hull Zodiac. We don't own a boat in Hawaii. We charter them. The funding for the work has been it's never been 5 years. I don't know that I'm going to have funding for the next 5 years. I know I'm going to have funding for the next year. So even though it would have been a great idea to buy a boat 20 years ago, or 15 years ago, or 10 years ago, or 5 years ago, I've never had the certainty to buy one. So we end up chartering boats. And one of the things we do is we build these bow pulpits on the front, and that allows a couple observers to get up high. It makes it a lot better platform for photography. These boats are good in almost any sea conditions, so you can leave when it's calm and come back when it's rough. We tend not to leave when it's rough. And effectively work over a pretty broad area. Now, we're studying a lot of different species and asking a lot of different questions. Some of the really basic ones are the species resident to the islands or part of open ocean populations. How are they using their environment in 3 dimensions? How deep are they diving? Are they foraging at night or during the day? Where are they going? What the abundance is of these different populations, and importantly, how are they responding to anthropogenic activities? And the main things we're looking at are either fishing activities or Navy sonar. Now I'll show a little video, and this is from an encounter we had with false killer whales off of Hawaii a couple of years ago. I'll mention a couple things before I start the video. It's taken from a GoPro on the head of one of the photographers, and you can see the camera lens, but these 2 black things on either side of the lens are green dot lasers. So they project 2 green dots onto the side of the dorsal fin, 15 centimeters apart, and you can use that to measure an animal. So they give us that added information of how big is the dorsal fin, how large is the distance between the dorsal fin and the head, so we can start to characterize whether they're adults or juveniles. Oops. And then you'll also see a couple other photographers in the boat. And this is a lone false killer whale. The fellow in the bow is holding an air rifle with a satellite tag on the end of the rifle. This false killer whale is approaching the boat. It's going to move over to the other side of the boat, and the very last frame will be tagging the animal. And I think at least one of the people in the audience is back. So there was the tag deployment. I'll pass around one of these tags. These tags, we have 3 different types of tags. I'm going to show you some of the information from it. This one has a dead battery. Normally it's worth $4,100. It's not really worth taking because it's a dead battery. It's 3 different types of tags. This one gives us both the location of the animal and how deep it's diving. We also have a tag that's worth $5,500 that also has a GPS unit on board. And then we have the cheap one, it's only $2,500 that just gives us the location of the animal. And the tags are really important because they allow us to look at what the animals are doing when we're not there, where they're traveling around the islands, how they're behaving at night. Last couple of years, we've also added drones to our toolbox, and we've been using drones for a couple different reasons. In this case, we're using the drone to actually collect breath samples from pilot whale. So when the whale exhales, we fly the drone through the plume of air and water droplets, and we can look at what's in the microbiome, so what's actually living in the lungs of the animals. We can also get DNA from the animals themselves. And then the other thing we're using the drones for are obtaining aerial images, and this isn't a great online, directly above one, where we can actually measure animals. And we can say that this animal is 2.3 meters long, and we can measure the width of the animal, and we can use that to not only say whether it's an adult or a juvenile, but how healthy it may be in terms of how robust it is. We do a lot of other things. We collect a lot of other information. We record seabirds because seabirds are an indicator of productivity. Anyone want to say what seabird it is? Probably David. Okay, David can say it.
Is it a Kermadec petrel?
It's a Kermadec petrel, yes. So we record. Sorry to put you on the spot. I should have put a harder one in. We record seabirds because seabirds are an indicator of productivity. When there's a lot of birds around, they're there usually because there's fish, or in this case, squid that the animals are scavenging on. We also collect squid and dead floating fish because they tell us something about what's available to the different species of dolphins and whales out there. This particular squid we collected on our last project in 2019. It's a new species of squid that hasn't been described yet. This is about the 15th specimen we collected here in Hawaii. It's Onapia is the genus, and that particular genus is only found, or was only thought to be found in really cold waters, and lo and behold, we're collecting all of these warm water species that are different morphologically. Now, in terms of our field work, I mentioned we come out to the islands, we charter boats, we go out in the water every day as long as we can, as long as the weather conditions allow. Our first project off this island was in 2002. Those yellow lines that you can see are the vessel track lines of where we went each day. In 2002, we did digital cameras weren't a thing then, so we were using film. So we only took less than 300 photos. They're a big change in 2003 when we switched to digital cameras. We're also collecting biopsies, which are used for genetic samples. And in those 9 days, we only had 4 species. But if you look at how close to shore we were, we're relatively close to shore. 2003, we came back for a little bit longer. We had got a digital camera, just one of them at that point, but we collected almost 3,000 photos, collected a lot of biopsies, had 7 species. And the reason why we had 7 species is because we were able to extend our effort further offshore, taking advantage of good weather conditions, and when the weather's good, we go as far offshore as possible. Next project on this island was 2010. We took 18,000 photos because by 2010 we had 3 digital cameras instead of 1 digital camera. And we had also added satellite tagging to our arsenal of tools. 10 species again, because we're able to get really far offshore, we're able to see many of the really deep water and sometimes oceanic species. So that's just what we've done off Oahu, but we've been working around all the main Hawaiian islands now for 21 years. Off Kauai, we've worked there for 12 different years. We just finished a field project there 2 days ago. Maui Nui, 8 different years. Off Kona for the last 18 years in a row. We have another Kona project planned later this year. And we have the least amount of effort off of Oahu of all of those. But we actually still know an awful lot about what's going on around Oahu for a couple reasons. One, I mentioned citizen science. We have 3 different companies off Hawaiian that send us photos pretty much almost every week of the year of what they're encountering, and that plays a big role. But also, as you'll see in a minute, many of the species that we're tagging off other islands, we may tag them off Maui or off Hawaii or off Kona, and they're moving among the islands. So we can say more about what's going on off of Oahu than just based on our boat-based surveys. Now, there's actually 18 different species of whales and dolphins in Hawaiian waters, and through our work and through work folks at HIMB and the Science Center, there's actually really strong evidence that 11 of these 18 species are resident to the island. So all the species that are shown here, there's actually resident island-associated populations. Not all of them are necessarily resident to Oahu, but most of them on this slide are actually resident to Oahu. And I'm going to talk about 5 or 6 of these different species. The first one I'm going to talk about are spotted dolphins, pan-tropical spotted dolphins. They get their name obviously because of the spots, although when they're born they don't have spots. Spotted dolphins are actually probably the most abundant species of dolphin around the islands in general. Although people don't necessarily think about that because they think of spinner dolphins being so close to shore, but the abundance of spotted dolphins is probably 5 to 10 times higher than spinner dolphins around the islands. Spotted dolphins acquire these spots as they age, but you can sometimes see they have these very complex spotting patterns, and in particular on this individual you can see sort of these starburst patterns, and those starburst patterns aren't really caused by sort of the natural acquisition spots as they get older. These guys and most other species of dolphins and whales in Hawaii get bitten quite regularly by cookie cutter sharks. And these cookie cutter sharks basically take chunks of the animals out that are about that big around and oval. And in the case of spotted dolphins, when those bites heal, like you can see right here, these lines, those are actually spots that occur right on the edge of one of these bites, and as the skin regenerated, we're basically dragged in towards the center of the line. So you get these really complex patterns. The other thing you can see on this individual is these linear scars up here, and this is probably an adult male. Male spotted dolphins have a lot more spots than female spotted dolphins about the same age, and this is probably from fighting with another adult male. So those are tooth rates from interactions with another spotted dolphin. Now, spotted dolphins in Hawaii are interesting for a number of different reasons, but one of the reasons is this is the species that fishermen often will seek out in order to try to catch ahi. Yellowfin tuna will follow spotted dolphin groups around, and fishermen have known that for a long time. Particularly off Kona, but it also occurs off of Oahu and a little bit off of Lanai. Fishermen will troll back and forth through spotted dolphin groups to try to catch ahi. Every once in a while, and we know this because fishermen have reported it, individuals will get hooked as a result. So we've been interested in trying to understand more about the dynamics of spotted dolphin populations in Hawaii. Part of that work, and I passed around that satellite tag, has been to satellite tag individuals and get an idea of their movements. These red lines around the islands are actually what NIMS has considered to be the boundaries of different stocks based on the genetics. The PhD study done a few years ago showed that the spotted dolphins off of Oahu were different than the ones off of Maui Nui, and different from the ones off of Kona, and the ones off of Kauai and Niihau are thought to be a pelagic population. So we've tagged a few spotted dolphins now to try to see, okay, the genetics say they're different populations. What are the animals doing in terms of movement? So we tagged this one off of Oahu in 2016, just 18 days, but you can see the really broad-ranging inshore, offshore movements. and crossing that sort of imaginary stock boundary. So very quickly demonstrated that there's a lot more movement within this than anyone thought, based on the genetics. We tagged another one in 2016. Again, this one was tagged off of Hawaiian, and only a 14-day period. Spent most of his time actually within the Oahu stock boundary, and then went on a little walkabout over north of Molokai and Maui and all the way to Kahala. So this kind of tag information gives us a lot more sort of real-time information of what animals are doing than the genetics, which are telling us about these long-term patterns. And I mentioned that the spotted dolphins off Hawaii are thought to be part of an open ocean or pelagic population, and the two different groups that we've tagged off of Hawaii have actually shown exactly that pattern. They spent very little time in the shallow water around Hawaii or Niihau, and instead spent most of their time out in water that's 5,000 meters deep, including coming over at least occasionally off of Oahu. So still a lot to be learned about this species around the islands. Now, another species of dolphin that again is resident to the islands that people are probably a little bit more familiar with, at least these are animals that you can see from the beach at times. They're inhabiting very shallow water, or bottlenose dolphins. And bottlenose dolphins, based on our photo ID work and the genetic work, again, this was the subject of a big genetic study that showed, in this case, there's a separate Hawaiian Niihau population, a separate Oahu population, separate Maui Nui population. And this shows satellite tag data from 12 different individuals that we tagged off Hawaii. And they really show a tremendous amount of site fidelity to Hawaii. It's always that 13th animal that really, again, an animal we tagged off Hawaii. It spent a bunch of time doing exactly what we thought it would do. And then it came over to Oahu and spent its time south of Oahu and towards Penguin Bay. This particular individual, we have photo ID catalogs. It wasn't intermingling with the Oahu resident dolphins, so we don't know exactly what it was doing, why it was doing this trip, but it just provides a very different picture into the lives of the animals. I wanted to mention, bottlenose dolphins are one of those species that do take fish from fishermen here in Hawaii. And this is a great example of the citizen science that I mentioned, a paper that we published last year with several of the people who are co-authors on that here in the room today, who are basically folks who spend a lot of time out in the water getting photos. These photos here, taken by Chuck Babbitt, are basically the first photos that we have of this individual that really suggested that it had been shot. And amazingly enough, it had been shot through the head and survived. Over here, this is an entry wound. It has all the characteristics of a gunshot entry wound. It's got inverted edges, and this is actually the exit wound here on the other side. Based on the angle of it, it was shot from above, went in high and came out low. And how this animal survived is amazing to consider. It must have just missed the skull and went right above the skull. But this particular individual, in these photos and in the whole photo series we have, was still being nursed by its mother. So it hasn't had to learn to hunt on its own. We don't know yet. We don't have any resightings of it in the last couple months. So we're not sure if it's still alive, but it survived an amazingly long period of time after the gunshot. And again, if you look at the affiliations of the folks, of the people who are co-authors on this, really it's folks who work on the water who have this opportunity to contribute. It's an amazing contribution. Moving on to, so now I've talked about dolphins, but now I'm going to talk about whales. And whales and dolphins, I mean, they're all actually oceanic dolphins that I'm talking about. Some of them just happen to be called whales and some are called dolphins. These are two species, both of which are found off Oahu, one of which is resident here and the other isn't. They're really hard to tell apart, so a lot of times when people see them in the field, they don't necessarily know what they're looking at. The bottom one, melon-headed whales, are often found in groups of 200 or 300. The top one, pygmy killer whales, are typically found in groups of 10 or 20, so that's often a good cue. Pygmy killer whales are one of the least known species of oceanic dolphins in the world. They were only ever first documented as live animals in the early 1960s, late 1950s. Prior to that, they had only ever been documented from skeletal specimens. And it turns out Oahu has a semi-resident population. We've tagged 2 different groups off of Oahu. This individual just down near Ko'olina, even though it shows it moving down the Penguin Bank and staying off of Penguin Bank, we know from the photo IDs that this is an individual and this is a group that is seen off of Waianae regularly, year after year, and appears to be resident to the area, and where it's resident to also just happens to include Penguin Bank and down towards Maui Nui. This is another individual that we tagged off of Oahu during that same field project in 2010. This one behaved very, very differently, moving inshore and offshore and inshore and offshore. Interestingly, this is not one of the animals that we see as resident to the area. And I'll just go back and you can see the first animal stayed in fairly shallow water, right on the edge of the slope, whereas the second individual that we think isn't resident was moving out into very deep water back inshore. We think this one's sort of just sampling the habitats. It's not an individual that's been in the area very often before.
Robin, why do they call it a killer whale?
Well, pygmy killer whales originally got their name because of the similarities in the skull and the teeth to killer whales. And, they were described based on a skeletal specimen originally, and they happen to have very large teeth relative to their size. So killer whales have extremely large teeth relative to their size. False killer whales and pygmy killer whales have very large teeth relative to their size. That's where the name came. They're also actually somewhat related to killer whales. They're along with pilot whales, but it really came from the description of the teeth. Speaking of pilot whales, this is another species of blackfish that's found around the islands. This is probably the most sexually dimorphic species of whale or dolphin in Hawaiian waters, although it really needs to be quantified. It really needs someone to do that. Maybe Shelby, who's over here, is actually probably going to take on that task. Shelby did a study with Cascadia a couple years ago, published, that basically quantified all the morphological differences between pilot whales and pygmy killer whales and false killer whales and mountain-headed whales. And one of the things from that study, through going through hundreds or thousands of photographs and measuring individuals, is the ability to quantify these age and sex-related differences. So, thank you, Sean. Good to see you. Now, pilot whales are actually the species that we see most often in our work. They represent almost a quarter of all the toothed whale sightings we have in our surveys. And there's a couple reasons for that. One, they're fairly common, but two, they're really easy to spot. They're large, they're slow moving, they spend a lot of time logging at the surface or very close to the surface. So we probably see them more often than we would a small, fast-moving species that goes on long dives that may be equally abundant. But we've done a lot of work with them, both in terms of tagging and biopsy sampling and genetics, photo ID catalogs. These animals are also homebodies. They're around the islands. They live here year-round. This is an individual that we actually tagged it off of Lanai, and you can see it spent a lot of time off Lanai, Penguin Bank, and around Oahu. This particular group appears to be basically resident to Maui Nui and Oahu. We have other groups that we've tracked that have had broader ranging movements, and these animals have been less frequently seen associated with the animals that we know are residents, and we use a combination of the photos and the tag data and the genetic data to really understand sort of the dynamics of each particular group. Here's a group that was tagged off of Oahu. You can see the little red dot there south of Barber's Point. And even though we know based on the photo ID that it's part of this resident population, it also moved broadly around Kauai and Niihau. And so it's a combination of all of these different types of data that we've been able to describe different communities of pilot whales around the islands. A western community that lives primarily around Kauai, Niihau, and western Oahu, central community around Maui Nui and Oahu, and they overlap, and then a eastern community off of Hawaii Island. Now, the last species I'm going to talk about, and it's my favorite one, you can tell because the stickers that I have up here are false killer whales. Please help yourself. They go well in water bottles, in cases of laptop computers. Bumper stickers on rental cars. False killer whales are my favorite species for a couple different reasons. They are, our work here with false killer whales in Hawaii is the longest term study of the species anywhere in the world, and it's the most well-known population of the species anywhere in the world. They had thought to be an open ocean species, and it turns out that there's not just one, but two different island-associated populations around the main Hawaiian Islands. False killer whales share a lot of traits with killer whales. So they were named false killer whales, as I mentioned, because of the similarity in the skull and the teeth. But like killer whales, they're very long-lived. The females live at least into their 60s, if not more. They don't start reproducing until they're 10 years old, probably for females. They only have one calf about every 6 or 7 years. And like killer whales and also pilot whales and humans, females go through menopause, they stop reproducing, and may live 20 or 30 years after that. And it's thought that menopause in all of these species really occurs because of the importance of grandmothers. Grandmothers play an important role. They know when, in the case of whales, they know when El Nino is coming and what to do, where to go. They know the long-term history, the long-term knowledge base of what to do when things are not happening normally in an environment, similar to elephants. So they share all of these features, and all of those features also put false killer whales at risk. It means they're very low reproductive rates, slow to recover from any sort of perturbation in the environment. Now, unlike all the other species that I mentioned, false killer whales also like to eat fish that we like to eat, or at least I like to eat. The upper right is an ono, opa, mahi-mahi, and a swordfish. And I don't know, does anyone like opa? They have a lot of overlap with what we feed on. They also eat jacks, the alua here, giant trevally. This is a list of known prey species of false killer whales here in Hawaii. This comes both from our observations and also the work of Christy West and others at the University of Hawaii looking at stranded animals. And so they're also an interesting species because they span from the reefs to the open ocean. So if you look at this list, it includes a lot of reef-associated game fish, like the aluas, and it also includes a lot of pelagic game fish, and bonefish even, as well as squid. And so there's a tremendous overlap between what they're eating and what humans are feeding on or fishing for. Now, if you've ever seen a flying mahi-mahi, it's probably because false killer whale here managed to capture that. We're really going for the mahi-mahi photos. Happen to knock it into the air. They also tend to be very elaborate in what they're feeding on. They like to throw their fish up in the air, particularly mahi-mahi. They actively feed during the day. Many other species like pilot whales are primarily feeding either really deep in the water or at night. And when they're feeding, they leap out of the water regularly, usually because they're trying to get mahi-mahi into the air. And so it's something that's easy for us to capture. Now, in our work, we've had a predominance of what we've documented from feeding on is mahi-mahi, but also mahi-mahi are really easy to identify. There's 4 or 5 different species of tunas around the islands, which, if you're a tuna expert, are really easy to tell apart. But if you get a quick glimpse of a tuna in the mouth of a false killer whale, it's usually very difficult to tell. So we think that they probably are feeding on a lot of things that we just can't easily identify.
Albert?
Yep.
I would think the mahi-mahi would be faster than the false killer whale.
Yeah, well, they're not as smart, though. And the false killer whales also cooperate when they're hunting. But what false killer whales do, and it's a case here where they, I think when they first detect a mahi-mahi, they they immediately try to stun it, and that's why they're hitting them, throwing them up in the air. And then once they've stunned it, the mahi-mahi seem to become even easier to play with, and then they spend a lot of time playing with them. And they do this more often with mahi-mahi than other species of fish. And other species like tunas, when they're hunting tunas, we never see them playing with the tunas. If they catch a tuna, they just catch it and eat it. Whereas mahi-mahi, I don't know, I think it must be a lot easier to catch than some of the other species. They also do interact with other species of fish. Can anyone tell me what species of fish this is? It's a crowned toby. Although we have seen false killer whales mouthing and eating straw fish as well. Yeah, so we know it's a crowned toby because we recovered it because they dropped it after playing with it. This was off of Oahu. Kim, were you on the boat for that? So crowned tobies, as many of you know, many of the puffers are toxic in some way. And you may have seen in the news a couple of years ago about bottlenose dolphins that were mouthing pufferfish and then changing their behavior afterwards. They basically are getting high off the toxins. Whether or not these ones were doing it for that reason, we don't know, of course, but they were mouthing it and then dropping it, and another one would mouth it and drop it, and they eventually moved on. We didn't really notice any difference in their behavior, so we don't know whether they got high from it or not. But, you know, they're intelligent animals. They know what's going on in their environment, and they probably have a good idea that mouthing a crowned toby might have those effects. So false killer whales, of all the species that I've talked about, also have the smallest abundance of any of these species in Hawaii. The estimated abundance is right now somewhere in sort of between 150 and 200 individuals for the main Hawaiian Island population. It's been the emphasis of a lot of our work, and we've tagged individuals that have moved among the islands. This is an individual we tagged off of Barber's Point 2010, and you can see in a month and a half it basically is just doing multiple rounds around the Oahu and Maui Nui, and a much greater degree of movements in a very short period of time, going back and forth and back and forth. And we've now learned that these islands here are really the core of the range of this population. Other individuals, again, this is an animal we tagged, same area in 2013, a little bit longer duration track. Some movements over to Kohala, but again, just regular movements around Oahu, around Maui Nui. I mentioned they feed on both reef fish and pelagic fish, which is also reflected in these tracks. Sometimes they're in very shallow water, sometimes they're in very deep water, unlike the other species that I've shown that tend to be either in one or the other. This shows basically the entire known range for this population. This line right through the middle of Hawaii Island is just because there were no locations in the 12 hours in between, but it basically went around the island. So they extend from east of Hawaii Island to just west of Niihau. And the area off of Waianae is a particularly good area to see these animals. It's not really an area where they spend a lot of time, but they go through it over and over and over again. So we had one case where we tagged a false killer whale off Waianae that circumnavigated Oahu 7 times in 20 days. So it just went around and around and around. And that exposes them to some interesting risks. For example, Barber's Point is the main place where oil is offloaded in the islands. And while they don't spend much time around Barber's Point, they go by it all the time. And so if there was an oil spill, it's a species that's highly likely to be exposed to it, again, with a very small population size. Now, the reason for the small population size is unclear, but we think the biggest culprit is probably fishery interactions. There's no observer programs in all the nearshore fisheries, but this is a species that is known in the offshore fisheries to regularly take catch from fishermen. In the longline fishery, take bait from the longline fishery. And there's a number of reports of them taking bait and catch around the islands as well. And we also see injuries, like you can see in these two individuals, from animals that have been hooked in fishing gear and have survived. So the animal on the right is, it shows it's missing a bunch of its lip, and it actually has a broken tooth where it's being hooked in the jaw, and basically the hook is pulled out and broken a tooth. The animal on the left was probably hooked in the mouth and was struggling against the gear, and that's why it has that injury on the leading edge of the fin. And in both cases, both of these individuals have survived this injury. The one on the left was one that we documented off of Waianae in 2017, and that individual was seen in 2018. It was also seen in 2019. So even though it has that horrific-looking injury, it was still able to heal. But this is another component of the work that we're trying to do, is document, particularly with these mouth line or head photos, evidence of fisheries interactions. And this information is particularly valuable when you combine it with information from strandings, work that Chrissy West and her colleagues at the university are doing, because when they get an animal in, they're able to look at what's in the stomach. Does it have evidence of, does it have hooks in the stomach? And there's been a couple animals that have had that. They're able to look in much better detail than we're able to see from these types of photos through the water of what types of injuries are in the mouth. And so it's been a really good collaboration with Christy and her crew to try to use these various lines of evidence to understand what factors are influencing this population. So I think this is my last slide, and I'd be happy to answer questions.
How have been does the vertical migration of humpbacks affect your documentation?
Yeah, so the question is, what does the migration of humpbacks to the islands, how does that affect some of the resident species? And it's interesting. I mean, humpbacks provide a tremendous influx of biomass to the islands. I think they have a huge influence on tiger shark populations and white shark populations because of humpbacks that are dying here or humpbacks that are getting predated here. We certainly see a lot of interactions between humpbacks and other species, but those interactions are more just cases where they're socializing, and juvenile humpbacks will socialize with pilot whales or with bottlenose dolphins or vice versa. But none of the species that we are really working with are killer whales do come from Hawaiian waters. They weren't on that resident slide that I showed. They're not part of the resident population, and they obviously are feeding at least occasionally on humpback calves when they're around the islands. But the other species are probably just using them as occasional social companions.
What's the status of the false killer whales on the ICUN list?
Yeah, so, well, I didn't say, so false killer whales here in Hawaii, this main Hawaiian island population is being listed as endangered. It was listed as endangered in 2012 in the United States. The IUCN listing was recently reevaluated in 2018 as near threatened. And near threatened is an interesting category. One of the reasons why it's near threatened rather than threatened is because there's so much uncertainty about the species, basically in almost everywhere throughout its range. It's the more research that's done on false killer whales, the more evidence that there's these small localized populations rather than huge open ocean populations. But because they're spending so much time in the open ocean where there's little in the way of observer programs. So the U.S. longline fishery, for example, has a really good observer coverage, but all the other longline fisheries that also co-occur throughout the range of false killer whales have extremely poor observer coverage. And so it's really hard for these open ocean species to be able to sort of quantify what influence things like fisheries are having on them.
Do you have any way of recovering those expensive tags? Or when you tag one, is it considered gone?
Yes, so the tags they're small in order to be able to fire them from an air rifle, which means they have no flotation on them. So they stay on, well, you saw some of the tracks were up in close to 200 days. We've had one on a false killer whale, 199 days. We've had one on a pilot whale here in Hawaii, I think for 220 days. But when they fall off, they just sink to the bottom. Yeah. And sometimes when we miss, we get them back. Other times when we miss, they sink to the bottom. So we just finished this field project on Hawaii. We had 5 tagging attempts. We got 1 on a pilot whale, 2 on bottlenose dolphins. We had one where the tag normally has darts on it, where the tag hit the fin of a bottlenose dolphin, but the dart didn't, and that one just sank. And then we have another one that missed entirely, and we got that one back. The one that sank was one of the $4,200.
How do you get them back? Do they have a line on them that you can pull them back?
Yes, so the arrows float, and they have a 2-pound test line from the tag to the arrows. So normally, when it hits the animal skin, the line breaks. But if it misses and it's calm, then the tag hits the water and skips, and then we can go pick it up. In rough seas, if it hits a wave, then it usually breaks and we lose it. Or in this case, because just part of the tag hit the fin, then that's when the line broke, came loose from the arrow, and so. It doesn't bother me, though. It used to bother me, doesn't it?
What do we have to do to get more whales to come by Hanauma Bay?
Well, I think if you set up a good spotting scope offshore from the bay, you would actually see a tremendous number of things. So one of the things I didn't mention, we've added a land-based component to our work the last couple projects. So we've had someone on shore with tripod-mounted high-power binoculars and tripod-mounted high-power spotting scope, and we haven't done it off of Oahu, but it's amazing what they're able to see from shore. Pilot whales, false killer whales, melon-headed whales off of Kona, spotting them from a kilometer inland, just with really good optics. There's a lot of things out there.
Traveling out there, we used to see.
Yeah, false killer whales. There is a great photo taken by a tour operator out of Honolulu of a false killer whale with the Waikiki skyline in the background. We've had a number of our tag false killer whales that have come right around Coco Head and right offshore here. And I think it just needs good binoculars and patience as well.
I have 2 questions. One is, how do you explain the discrepancy between the tagging and the genetics? Do you think this is culture and tribalism, where they're only interbreeding with, their own culture, tribe, and they each have like different versions of language? And that's what explains it.
Yeah, well, and false killer whales are a great example. I mentioned there's another insular population of false killer whales in the northwestern Hawaiian Islands. And we know that the northwestern Hawaiian Island population and the main Hawaiian Island population actually overlap. They overlap with Kauai and Niihau, but there's been 2 sightings of the northwestern Hawaiian Island population off of Waianae as well. So they overlap, but from the genetics, well, we've never seen them together. The photo ID has never shown them together. And the genetics show that they're not interbreeding. And I think that it relates to tribalism. I think is a good way to describe it. These are long-lived animals that know everyone in their community. It's a community of less than 200 whales. They've interacted with those 200 whales for their entire lives. If they can easily tell each other apart from sounds they make. It's much harder for us to tell them apart from sounds, but I think that they could probably detect another false killer whale 5 or 10 kilometers away, and I would be willing to bet a lot of money that they can easily tell that those are not part of their community. Then it gets to, well, why don't they associate between communities? It's interesting from the genetics. So there's 2 different false killer whale genetics are pretty well understood around Hawaii. There's mitochondrial DNA, which tell us about their maternal lineage, and then there's nuclear DNA, which tell us about interbreeding between populations. And the maternal, well, I didn't get into this, but false killer whales, almost every false killer whale in the main Hawaiian Islands has the same mitochondrial haplotype. They're all related to each other from one colonization event that probably happened tens of thousands of years ago. The northwestern Hawaiian Islands population has that same mitochondrial haplotype. So they're probably all part of that original colonization event. And when you compare it to other species of whales, pilot whales, pygmy killer whales, bottlenose dolphins, all of those have dozens of haplotypes. So there are multiple colonization events around the island. So false killer whales are different in that way. But when you look at the nuclear DNA, the main Hawaiian Island false killer whales and the northwestern Hawaiian Island false killer whales, even though they originated from the same event, are not interbreeding. Or if they do interbreed, it's once every few hundred years at most. But the pelagic false killer whales have a completely different habitat. They're not only different habitats, but they're not closely related habitats. And the same type of comparison with nuclear DNA has shown that the pelagic false killer whales are not interbreeding with the main Hawaiian islands. But the pelagic ones are actually interbreeding a little bit with the northwestern Hawaiian island animals. And if you remember back to one of the first slides I showed, if you looked at that productivity, the productivity around the main Hawaiian Islands is much lower than the productivity in the northwestern Hawaiian Islands. And I think that means that in the northwestern Hawaiian Islands, two things happen. The pelagic animals sweep back and forth through the northwestern Hawaiian Islands and are more likely to interact with the northwestern Hawaiian Island animals. And the northwestern Hawaiian animals may be roaming over a broader area because it's a higher productivity area in general. So, long answer to a short question. You said you had two questions.
Yeah, the other one is, any observations or evidence of any of these different species associating with whales to take advantage of placenta for humpbacks?
Yeah, we have not ever seen any of the evidence of that. I mean, certainly during the winter, when humpbacks are here, you see a lot of interactions between odontocete and humpbacks. But most of those interactions are with juvenile humpbacks, in my experience. The prolonged ones, like off of Kauai in particular, because humpbacks regularly move between Kauai and Niihau, and so we see humpbacks out in deep water there more so than we see the humpbacks in deep water off other islands. And when humpbacks are in deep water between Kauai and Niihau, we regularly see pilot whales with them, we see rough-toothed dolphins with them, we see bottlenose dolphins with them. But it's often juvenile humpbacks that are really just hanging out with them. It's certainly possible, but
I was curious about the genetic diversity of the populations that you're seeing. Are you seeing changes over time in those? And do you feel like you have like a really solid representative sampling of all the residents?
yeah, so for genetic diversity, we don't really have any way of looking, at least in the short term, of changes over time in genetic diversity. False killer whales, so one of the things that the geneticists, and I'm not a geneticist, so don't quote me on anything I say related to that, but I can put you in touch with some. The geneticists basically estimate effective population size, which is a long-term measure of the number of breeding individuals in the population. And for a population like the main Hawaiian Island false killer whales, that effective population size is only about 60 individuals. But I think it would take, because it's a long-term measure, I think it would take longer than our lifespans to really start to see a change.
Question.
Any resident sperm whale populations?
Now, sperm whales are interesting. I didn't mention them as part of the resident group because we don't have any evidence that they're really resident. We have tagged some, and when we tagged them, they've moved over broad areas offshore of the islands. That said, there is at least one individual that's been photo identified off of Kona in 3 different years over about a 10-year span. So I don't think they're resident, but I think they might come back through the islands on a semi-regular basis.
How do you take a biopsy?
We use a crossbow to take a biopsy. So it's a skin and blubber biopsy. We use a dart that is 8 millimeters in diameter and about 15 millimeters long on a crossbow bolt that has a float that also acts as a stopper. So we basically shoot it, it hits the animal, bounces off, and falls into the water, and then we go and recover it. And those biopsy samples we use for a whole variety of studies. 1/3 of the skin gets sent to a genetics lab for looking at the sex of the individual and the mitochondrial DNA and depending on what species, other genetic studies. 1/3 is going to a lab in Florida that's looking at stable isotopes to look at trophic ecology. The other 1/3 of skin actually gets archived at University of Hawaiian Christie West Lab, sort of for any long-term use that we haven't thought of yet. And then the blubber, most of the blubber from most of the species is actually going to Christie as well, either for some blubber histology work, and I think there might be someone here in the audience who could talk about exactly what that's about if you're interested. And then we've also used some of the blubber samples for toxicology as well. And Christie is also archiving them for potentially for hormone chemistry to look at either reproductive hormones or stress hormones. So there's a lot that can be done with one little tiny biopsy sample. And we're trying to partition those samples so they can be used for as many different things as possible.
Have you noticed any differences in strategies between the population of pelagic and your reason to believe that that's locally transmitted?
Yeah, it's a good question about whether pelagic false killer whales and insular false killer whales are feeding in a different way. And unfortunately, we've had relatively few encounters with pelagics. When we have seen them, we've seen them feeding on the same species of fish, well, the same pelagic game fish that insulars have been feeding on, opa, mahi-mahi, and ahi. We have a much smaller sample for pelagics, so it's hard to say. One of the things that we would like to do is look at stable isotopes between pelagics and insular. So we have a manuscript that's just about to be submitted on toxicology and insular false killer whales. Actually, it includes pelagics as well, and it includes a bunch of stable isotope results for insular false killer whales. And I never mentioned, but another similarity insular false killer whales have with killer whales is they live in these very long-term stable groups that are similar to killer whale pods. We call them clusters in false killer whales, but there's evidence that the different clusters are feeding on different things based on the stable isotopes. So one of the clusters has no overlap in stable isotope values for carbon, but overlap with nitrogen, and the other ones have no overlap for nitrogen, but overlap in carbon. And part of what we need to do, and if any of you are fishermen and are willing to collect little cubes of fish tissue for us, please talk to me afterward. Part of what we need to do is start to build up a sample of fish samples of a number of different species to be able to put the false killer whale samples in context. And we have a big stable isotope project that was just a very false killer whale specific project, but we have another big stable isotope project that's looking at stable isotopes in about 12 different species of Adonocetes in Hawaiian waters through Florida International University. And for that project, we're actively working to get a number of fish and squid samples for comparison to try to put it all in context.